contributions. So with that, my name is Jason Karamchandani. I'm a neuropathologist and a clinician and an associate professor in the Department of Pathology here at McGill University. The reason I'm talking to you today is in my capacity as the director of the CBIG repository. This is the Neuro's open science combined biorepository and patient registry. Now, the motivation for this is clear. This is a real sample from a 7-year-old boy in Texas. This is a picture sent to me by one of my training colleagues, Dr. Krishnan. And this patient wasn't feeling very well, so went to an emergency room and had a blood draw. Within 45 minutes of being in the emergency room, a pathologist, so my kind of doctor, looked at this blood film and made a diagnosis of acute leukemia. And in fact, just with the presence of these little hour rods, they were actually able to specify that this is an acute myeloid leukemia, and were able to immediately start a targeted treatment that helps the survival of these patients. It's a wonderful example of medicine succeeding. In 1950, if you walked into a hospital and were diagnosed with acute leukemia, you died. Everyone died. Leukemia was actually the disease that allowed for the creation of the concept of medical chemotherapy. Dr. Sidney Farber used an anti-metabolite in patients, and it actually improved patient survival. Since then, every decade, there has been an addition to the armamentarium of clinicians who treat this disease. And the survival rate for leukemia in 2023 is between 80 and 90%, depending on the subtype. It's a huge win for medicine. If we compared that to neurological diseases, we are not doing as good a job. So we started with treating Parkinson's with L-DOPA in the 50s in animal models and 60s in patients. And since then, there have been no really significant medical therapies to take care of this currently incurable disease. That's true of Parkinson's, Alzheimer's, ALS, stroke, and many neurological diseases. So the CBIG repository was born out of a sense of urgency. And our goal is simple. We exist to accelerate the development of meaningful treatments for neurological diseases. One of the main hopes of this initiative is it also empowers patients by allowing them to participate in their care. We had an open science symposium here just last week, and one of the CBIG patients actually moderated one of the sessions. And I really enjoyed what she mentioned to the group, which is that this participation made her feel like she was taking control back. So she felt helpless, but now she was doing something. And in fact, she's organizing an initiative that we'll talk about later today. CBIG is an acronym. It stands for Clinical, Biospecimen, Imaging, and Genetics. This is the different kind of stuff that we collect. Basically, we collect things. Those would be the biospecimens, so bits and pieces and liquids and stuff, and different kinds of data. And our goal is to share this as openly as safely possible. And we're able to do that thanks to the work of Samir and his team. The who is basically everyone. We can consent adults, pediatric patients, patients who need the use of a substitute decision maker, as well as healthy controls. And depending on the disease and the workflow, we can either use pre-consent, meaning the patient signs the consent form before we obtain stuff, or post-consent. And for different clinical scenarios, we do whatever is considered ethically the higher standard. This is a long list of some of the stuff that we can take. Obviously, blood and derived products. But some of the unique things we have access to is because we're at a combined academic center, but also a hospital here at the Neuro. So we have neurosurgeons and operating rooms and access to brain tissue and skeletal muscle, cerebrospinal fluid, and peripheral nerve. These are kind of samples that are very difficult to find in most environments, and it's one of the things that makes this project unique. I know Samir is going to give you more details about some of this, but the combination of stuff and data is what makes this powerful. So we obviously have longitudinal clinical information, so how a patient is doing through time, but we also have access to both research imaging as well as clinical imaging, and you'll hear more about that in a bit. The G in CBIG stands for genetic data, and we have a tight integration with the neurobioinformatics core, and we now have thousands of patients, more than 20,000 actual individual data elements from thousands of patients in this cohort. And that includes things that range from whole genome sequencing to limited panel testing, depending on the patient's disease. And we have the ability to interrogate patients' chart as well as their electronic medical record. I don't know how many of you are based here in Quebec, but we all, with a RAM-Q card, have a Dossier du Santé du Québec, which collects a certain amount of clinical information. At the very minimum, it's the drugs that we take, our lab values, and our imaging results. This was the original goal. So we wanted to develop new tools to allow us to study human neurological diseases by consenting patients, taking their blood cells, turning them to things like stem cells, and then studying those cells in a dish. And these cells are particularly from patients with genetically caused disease. So patients with a mutation that causes Parkinson's disease or a mutation that causes ALS. And it worked. So this was the first time I saw this was in 2017, and I just about jumped through the roof. These are what we affectionately call mini brains. These are three-dimensional organoids derived from patient cells. So the blob on the right of your screen is brown because these cells are producing neuromelanin. These are dopaminergic neurons. Those are the cells that are actually implicated in Parkinson's disease. So now for the first time, we're able to study human neurons from patients with human disease and perturb them, put them under different conditions and see, can we return these cells to their normal phenotype? And I'm a pathologist, so I can't help but show you what it looks like under the microscope. And you'll just have to trust me. These pink fluffy cells over here are differentiated neurons, and the bluer regions at the top of your screen are just slightly less differentiated. So you can see this is a brain in maturation. This is an example of part of the catalog we have of the patient-derived cells that we now share as openly as possible with scientific partners around the world. We now have more than 160 of these patient-derived cell lines. And you can see here that we classify the genetic alteration and the genes that are, and the disease that are involved. Most of these are related to Parkinson's disease, but we also have some ALS lines here. These mutated lines are paired with an isogenic control. So when you compare two different patients, obviously we have different genotypes. With an isogenic control, to the best of our ability, you are comparing cell lines with one difference in a relevant mutated gene. And all of these cell lines go through an extensive quality control process. And with Samir's help, we're actually putting this in the database as we speak. So where we are now, we started in 2016, collected our first patient in 2018, because it took us 18 months to get the approval from ethics to proceed with this. And we now have, this is out of date, it says 30,000 samples. We have about 75,000 samples from 4,000 patients and healthy controls. Our largest cohorts are in Parkinson's disease, ALS, neuromuscular disease, and a growing cohort in multiple sclerosis. We also have lots of small individual patients with very rare neurologic disease, and I think there's particular value in aggregating cases from patients with rare diseases. This platform has become the hub of many pan-Canadian initiatives. So there's 3 initiatives listed here, CalSNC Capture and its successor Capture 2. All of these use the CBIG platform as the core hub for the specimen processing, and for ALS, the data hosting as well. That's also true for Parkinson's disease, for the Quebec Parkinson's Network, and the larger Canadian Open Parkinson's Network. We are also the hub for the Pan-Canadian Neuromuscular Network, funded by the CHR, NMD4C. And there, I'm very happy to say, when this network was renewed in 2023, open science was moved from essentially non-existent. It was not mentioned in the first version of the network to a core cross-cutting theme for the renewal. And that's a wonderful example of this sort of polymerization or propagation of the concept and philosophy of open science. And similarly, we're expanding into autism and intellectual disability. So I just last week was asked a question by people from France, the Pasteur Institute. How do you know if what you're doing is working? This is one metric that I like to use to show that this is having an impact, because we had 3 collaborations in our first 3 years, and we had 40 in the last year. So we've now completed more than 90 collaborations with academic and industry partners. And these span from people here at McGill or Université de Montréal to University of Toronto, Harvard, Stanford, Oxford. And we also partner with industry. So that can be anything from small biotech startups to enormous pharmaceutical companies like Merck and Takeda. All of these collaborations are conducted in the open, meaning that some of the data that's generated with the experiments by these outside groups is returned to the CBIG collection, and that data is made available to subsequent investigators. A nice example of that would be some RNA-seq work with collaborators at Takeda in Japan, where they did RNA-seq on 50 samples from patients with ALS. That costs about $2,000 a sample. So that's $100,000 worth of scientific data that is now accessible to researchers here in the province, across the country, and around the world without having to spend that significant amount of funding. So that's one of the goals of open science, is to reduce duplication and make things more efficient. So we've collaborated with partners all around the world. All of this required robust ethics and governance. I told you it took 18 months for the first submission. It took 2 years for us to be allowed to include children as part of this cohort. And we did so at the request of our scientific collaborators. Autism and intellectual disability presents in children. It doesn't present in adulthood. So we had to go back to the drawing board and start again. I'm going to skip over this, and I'm happy to answer any questions about this, but we did have to develop a bunch of new tools. The open transfer agreement, which allows us to execute collaborations in as little as 4 days, with both sides having sign off on a legal agreement, is really remarkable in that it strips out all of the language that universities love around the development of intellectual property. So if we share something with a company and they make an invention, they don't owe McGill anything. That's shocking to administrators, and sometimes people get a little unsettled by it. But one of the core core principles of open science here at the Neuro is that intellectual property at the level of basic science slows things down. There is a role for intellectual property. We don't dispute that, but it's closer to the commercialization stage of things. And at this academic center, we're focused on more of the basic concepts in molecular biology. We have a governance structure that meets twice a year and looks at all of our activities. And this is composed largely of external members with expertise in law, ethics, biobanking, databasing, as well as internal members, including a patient representative. And this structure is meant to ensure that we are being true to the initial vision of the project, and that we are holding ourselves to the best practices and standards of outside projects doing similar things. Although to this date, we are the only open science repository like this in existence. We'll have some partners in the coming years, I hope. So when I say open and we're talking about patient data, that's obviously not that everything is out on Google. There is sensitive information, including genetic information. So we have this concept of tiered access. So the things that can't harm anybody is made open, metadata. Where there is individual patient level data, we require our users to register. So we know at least who's looking at the data, what they're looking at, and all of that is tracked with Samir's incredible Loris database. And finally, there's controlled level access for stuff like cerebrospinal fluid or brain tissue. That's non-renewable. So we have to make sure we're using it to the highest scientific use possible. And so those requests are reviewed to a tissue and data committee, which remarkably turns around decisions in 48 hours. So I want to make sure that we have enough time for Samir to cover the very real discovery for me that this project, which I thought of as being really centered around specimens and stuff, was really an informatics project, because none of this is real unless you can share these materials and data with other investigators within the university, within the province and country, and even more successfully around the world. And for that, we use the LORIS database. We're in the process of validating a new virtual consent platform, which I think is really exciting, because we all lived through COVID and people don't want to come to a hospital if they don't have to. So this new platform allows patients to consent where they want, when they want, on the device of their choosing, and at their own pace. And that was based on feedback from our multiple sclerosis patients, some of whom mentioned that it was cognitively challenging on them and draining to try to sit through the consenting process in one sitting. So we're making some changes. I know some of the earlier speakers talked about EDI, and here I want to confess a failure. We did not think about EDI when we launched this platform in 2016. Fortunately, we've taken this opportunity to rethink and reintegrate EDI initiatives into the CBIG platform. So one of our deliverables for 2024 is a brand new demographics instrument that was built in coordination with the Neuro's EDI committee. And with Samir and his team, we'll be implementing that in the coming year. I believe you're also platform, which is a wonderful way for scientists to discover and analyze existing and mature data sets. I mentioned we're working on the virtual consent. And thanks to Samir, we have a new workflow for how to interface with the clinical world. And so there's now a server behind the MUHC firewall that pulls MR scans from our patients, de-identifies them, and then transfers them to our research database, which Samir will tell you more about. So I'm happy to take questions after Samir's presentation, and I want to thank you for the opportunity to speak with you all today. With that, I'm going to pass it off to Samir.
Right. So I'll sort of transition a little bit, partly to MRI, and partly I won't. I'm very much academic, so I will do my best to show how this goes beyond the academic world. Can you see my presentation? I just want to make sure. Okay. So first of all, I do have a disclosure. I'm part of Lasso Informatics as one of the shareholders. It's a private company. It's the only disclosure I have, I think, other than being part of LORIS, which is not a private company, so not much to disclose there. LORIS is the underpinnings for CBIG, so I won't spend too much time on showing you what Jason just showed you, which is the repository. The few points to highlight is that there's a lot of imaging data in there, even open, if I'm not mistaken, definitely registered. So we're allowed to release MRI data. It wasn't an easy process, but that is there. As you can see, there's some numbers right now. They're changing all the time, and so there's much more data in the repository. This is sort of what it looks like. Here's sort of how it's all bridged together, multimodal data, including MRI data, included in the repository. Just a quick view. At the top, you can see that there's different kinds of even domains of modalities, imaging, behavioral, biospecimen, epigenetic, EEG these days. I mean, for those that are into MRIs, EEG is also something that is gaining a lot of traction in the imaging world and has been around for a long time. I could probably do a whole talk just on that one. But this is what our MRI browser looks like. We have some developed tools. When WebGL first came to browsers, we decided to dive into the not just MRI curation, but also MRI visualization. So we have surface viewers, we have volume viewers, all kinds of different viewers, where in this case, you can, look at your cortical thickness by overlaying the cortical thickness files on the surfaces that are generated. So you can see different views of this. There's annotations involved in this as well. So it's a pretty rich tool. It's called Brain Browser. It's just part of the suite of tools that we have that are all open source, for the record, and available to whoever wants to leverage that. One of the key points as I transition through this is, how do you get all the data? So we have built something that's 20 years old now, but has gone through multiple iterations, called the data query tool. If you wanted MRI data, you can log in there right now. Go to the CBIG repository. You can get MRI data at your fingertips. There's a whole bunch more coming. Like Jason described earlier, we're in the midst of trying to get access to MS data, not just MS data that's being collected now, but a huge history of data, which is very rare. Most of the times you can't find old data. So hopefully we will be in a very short time frame, giving the world access to possibly the largest amount of MS imaging data and MS data openly available. So there's that. This is what the query tool looks like. There's many ways where you can actually, not just get your MRI data as a file or a CSV file, but you can actually also submit it to other environments like high performance computing environments, which we've also specialized in over many, many years. So rather than getting stuck on your own server with whatever processing environment you're in, if you wanted to do this in scale, you could easily link to high performance computing environments. We are offering all of these solutions to end users that may not even be that adept in HPCs, or maybe even be supercomputer geeks. So our goal with CBrain, Loris, all of these pieces of software are to allow for anyone. You may know about how to do image processing, but you may not be so strong in the computing side. So we want to make this as easy as possible. So that I just highlighted very quickly the CBIG ecosystem and how it's underpinned by all sorts of other software. But I wouldn't be doing due diligence if I didn't share some of the challenges in sharing especially clinical MRIs. So Jason already touched on this, where the ethics, legalities, and privacy is very, I think that's your starting point. If you can't even ethically share the data, then that's difficult. We have delved into the world of even publishing how to do registered access and having consent codes, perhaps, which is important, especially as you go through different jurisdictions. GDPR is different from HIPAA in the States, which is different from Health Canada's compliance. So these are all very important starting points. As Jason mentioned, we have an e-consent module built in to our world, so that this alleviates the complications of sharing data that way. But just to go back to this, like the one thing that is clear to me, this is a long journey for me, the legalities are very different sometimes from the ethical restrictions of wherever you're at. The ethical restrictions are usually our IRB or REB boards, and they are not always necessarily up to speed on the legal, not just restrictions, but what's allowed. So in many cases, and we've worked very closely with lawyers on our side to share, for example, MRI data, and they're not like, sometimes hospital environments are too restrictive. They don't want to take any chances, but they're not actually barred from sharing the data. So there's a whole world into this. I won't necessarily spend too much time on it, but one thing for sure, I know the first speaker was asked, like, does open science mix well with all these environments, right? And or like the tools, sharing tools. And sometimes it can be complicated. So going through getting accredited, for example, in the States, FedRAMP accreditation is not necessarily the fastest process in the world. GDPR, you see like all the chapters there of what might be required. So why, like, privacy is absolutely extremely important from my point of view for everything we do, but so is, you know, accelerating scientific discovery and getting data out to people. So it's bridging that gap that's very tricky. In our efforts to help, at least wherever we can, we've created all kinds of software. So there's something called DICAD. It was a lot of stuff in my world starts off at hackathons and actually turns into real software. So DICAD's just for anonymizing DICOM header data for imaging scans. So, you know, it's a standalone tool, open source. Feel free to use it wherever you want. This helps because you know, we can whitelist or blacklist certain DICOM header fields that are identifiable. We've done the work. We have huge lists of, you know, what our defaults are. So handing this off to researchers, I think, makes a difference. Another couple of key points that are important for sharing data is, you know, first you have your ethics so that you can actually share it. And then if the data is not standardized, well, that's an issue. Matthew mentioned something called BIDS, which is the brain imaging data structure. We have, that's also started, by the way, at a hackathon. And this has been a long journey. It has actually taken over the imaging world. I did not expect that at the beginning when we were all, I've been part of many different little initiatives like this. You never know which one is going to take off, but this did. Another point to mention is that I'm the chair of the INCF Best Practices and Standards Committee, very specific, but the idea is, and we go through a whole vetting process all the time, we're trying to make proper best practices and standards shareable to the world. And a huge amount of the focus is on MRI or imaging data, but it goes well beyond that. So this is another key point, and that includes not just the raw data, but you know, provenance information, quality control, annotations, derived data, you name it. So that's there's a whole world onto that, and if you're not standardized, you're never going to be able to share MRI data properly. So just to kind of get towards the end of this particular presentation, I want to also talk about different kinds of perspectives. So I do research. I am a computer programmer, but I'm also kind of a participant. I don't know if I go as far as say patient, although I have been, but I've had a lot of MRI scans done. So my brain's been scanned for almost 20 years now, and if I'm not the most scanned person in the world, I'm definitely one of them. And so I've released all that data on the Canadian Open Neuroscience portal. You can go find it here. And also I've spent the last 5 years as a subject for an AI study, getting scanned 3 hours a week. So there's a huge amount of MRI data there. Collectively, I'm a little worried that I have so much data that somebody's going to want histology on my brain, but we will hopefully stave that off for some time yet. The point being, though, is with these open science portals, you can share your data. Even if you have one scan of your brain, you know, there's like sharing is sharing. So like I said, there's sometimes complications with the ethics, the legalities, the standardization, but we're doing our best to make this all available. I'll close by also just, you know, not just challenges, but there's a lot of benefits in sharing MRI data or any kind of open data to the world. There's often a perception that if you share, you're going to get scooped. And that does happen once in a blue moon, for sure, and maybe more. But that fear, I think, has driven the community for many years not to share. But the reality is, what I'm seeing is those who share get a tremendous amount of exposure. They have way more collaborations. They actually get more citations by usually, you know, you'll create the first initial paper, whether it's a white paper on, you know, the methods of what you're sharing or the data itself. And then if you share, everyone cites your white paper or your initial citation. So there's really, like, I think the fears are while they do happen, they don't happen nearly as much as people worry about. So, there's less money spent, there's tons of opportunities. It definitely improves reproducibility in our world. And so our goal is to enable scientific discovery. And I have this strange feeling that deep learning and machine learning technologies are like, they're hungry for information. And I think they're going to drive even more sharing and openness, particularly on the MRI side of brain, because it's patient data, so it's always been a bit tricky. Couple of quick examples of MRI data that's shared with the world. Prevent AD, this is a 10-year study on preventing Alzheimer's. So they look at people who are at risk for Alzheimer's or before they actually get it. And it's a longitudinal study. And this data went through the painstaking measures of reconsenting everyone so that it could share the data more broadly with the rest of the world. So I want to draw some attention to that one. Final point is it's also about community sharing. So BrainHack has been a kind of a grassroots thing that has sparked a great deal of interest all across the planet. I've been there since the beginning. It was just a bunch of geeks sharing MRI data or methods at first, but now it's really expanded beyond. And of course, we always like to have fun. So there's galas about open science where all this stuff is discussed over beer, and sometimes these things actually get us into trouble. People pick up on this and actually want to know more. So the things like this free beer and free speech conversation got actually a lot of notice by some people. So it resulted in all sorts of more presentations and conversations. So with that, I think I just want to thank everyone, and we're open for questions between Jason and I. Happy to answer whatever anyone has.